Greetings of the day. Myself, Dr. Ranjana Bengani from the Department of Aquatic Biology, Veer Normal, South Gujarat University, is thankful to the organizer for giving an opportunity to present a paper on third virtual international conference on advance in food and agricultural science and technology. The paper entitled Unlocking the Potential of Clove Plant Leaf Extract as an Antimicrobial Tool for Fish Pathogens is an important research that has been carried out at the Department of Aquatic Biology as an important agent for a fish pathogens. So fisheries sector in India is a booming sector which has brought a blue revolution in India and has provided an inexpensive source of protein. Aquaculture in India plays an important economical role. The share of inland fisheries and aquaculture has gone up from 46% in 1980 to over 85% in recent years in total production. So this fishing industry requires a lots of handling and storing, transportation, distribution, marketing, and selling of the fish or fish product. During this process, the fish is being passed through a different series which prone the fish to get infected. So this fish which may get infections from different bacterial and viral agent, what will be the inexpensive and organic source that can treat these pathogens. So as India is the second largest producer of the fish in the world, so keeping in mind the research was carried out to use the clove leaves as a source of essential oil as an antibacterial agent for the fish pathogen, as it has been revealed that clove blood oil have lots of antibacterial and antiviral properties that makes it very important essential oil. So and eugenol is the chief bioactive constituent of the clove plant. So clove blood oil has antibacterial activity, antimicrobial activity, anti-diabetic activity, antioxidant activity, insecticidal activity, anti-inflammatory activity. So these lots of properties that are found in the clove blood oil. My aim was to know that such antibacterial property is present in the clove leaves also. So considering the importance of clove blood oil as an antimicrobial agent, present study was conducted to investigate the potential of oil extracted from the clove leaves as an antimicrobial agent for fish pathogens. The study aimed to find a better and cheaper source of eugenol for better performance in terms of development and survival of fish. As different literature was surveyed, lots of study was carried out regarding the antimicrobial property of the clove bird oil, but very little study has been conducted in this aspect. So this study will explore the potential of clove leaves as a good source of eugenol and its effect on infected fish. So to fulfill the aims and objective of the study, fish sample were collected from the fish market that was infected, and from the infected fish sample were prepared for the isolation of the microorganism from the fish, and from the isolated microorganism qualitative analysis was carried out, like different types of biochemical test and gram staining test, in order to find out the genus of the microorganism that has been extracted from the fish sample. So next step was the collection of plant leaves. Clove leaves were collected. It were air dried under the fan. After that, powder were prepared, and prepared powder was used for the maceration extract. And volatile oil was separated, dried over anhydrous sodium sulfide, filtered and stored in dark brown water prior to use for the antimicrobial activity. Analysis of extracted volatile oil was done by use of GCMS technique. Identification of different types of chemicals were done. antibacterial activity of clove leaf essential oil was carried out. So antimicrobial activity assay include cup plate method and Dick's method. In the cup plate method, nutrient agar plate was inoculated with the test organism with a depth of 4 to 5 millimeter. Then it was allowed to solidify. Then nutrient agar plate was divided into 4 different proportions, and with the help of sterile borer, 4 cavities, one in each portion was made. Then 100 microliter, 50 microliter, 25 microliter of leaf extract were filled in the cavities, and in one standard antibiotic solution was filled for positive control, and the plate were incubated for 24 hours at 37 degrees centigrade. This diffusion method was also carried out, in which nutrient agar plate was inoculated with test organism with a depth of 4 to 5 millimeter and then allows it to solidify. Then with the help of a sterile forceps, antibiotic discs were placed slowly above the plate. Then plate was incubated for 24 hours and zone of inhibition was measured. For determination of antibiotic potential, a commercial antibiotic discs Icosa high media was used. These discs contain the standard antibiotic named amikacin, tetracycline and ivaprim. So these standard antibiotic were used in order to know what is the inhibition zone of this antibiotic with respect to the inhibition zone of the clove leaf essential oil. Against the selected antibiotic individual microorganism, E. coli and Staphylococcus aureus, which were isolated from the fish pathogens, were used. The zone of inhibition given by the standard commercial antibiotic were compared with the zone of inhibition of selected leaf extract. These are the result in which E. coli and S. aureus that were isolated through different biochemical tests. And antibiotic activity of leaf extract were measured. E. coli in 100 microliter, 50 microliter and 25 microliter, and Staphylococcus aureus in 100 microliter, 50 microliter and 25 microliter, and 3 replicate tests were carried out and inhibition zone was measured. So zone of inhibition for 100 microliter for E. coli was 90 millimeter, and for S. aureus was 20 millimeter in the leaf oil. Similarly, amikacin 30 unit, it was 22 millimeter and 26 millimeter, 19 millimeter for tetracycline for E. coli and 28 millimeter for S. aureus. For ivaprim, it was 35 millimeter inhibition zone and 18 millimeter inhibition zone for S. aureus, and clove bud oil that has 22 millimeter inhibition zone for E. coli and 26 millimeter inhibition zone for S. aureus. So from the result, it can be concluded that the leaf oil has a very comparable inhibition zone with respect to amikacin, tetracycline, ivaprim and clove bud oil. So this leaf oil can be used as a cheap substitute against different types of expensive antibiotics which has different types of side effects. The present work aims to identify and isolate pathogenic microorganisms from spoiled fish. Spoiled fish were collected from the market and samples were collected from the abdominal and fin region. The study of clove leaves reveals that this extract can be utilized to formulate drugs and further utilized as medicine from natural components against pathogenic microorganisms. Thus, for the new researchers to investigate the molecules responsible for antimicrobial activity, natural product can be a good substitute against the expensive antibiotics. So standard antibiotics such as tetracycline, ivaprim, amikacin, which are broad spectrum antibiotics, can be replaced by eugenol, which is a chief bioactive compound of the clove leaves. For commercial practice in industry, large scale of work, cost, effectiveness and concentration always make difference in business as well as marketing perspective. So with this concept, maceration technique can be used for methanol extraction of eugenol. Clove leaves extract can be used as preservative for the storage of different types of fish byproducts and products. Thank you for your patience and attention.